Welcome back, everyone. Dr. Linder here. We are continuing with muscle physiology. Let's look at the contraction and relaxation of the skeletal muscle fiber. Remember that sliding filament mechanism or that sliding filament theory of how actin and myosin, those two contractile proteins, interact? Myosin pulls on actin and it causes that thin filament, right? Actin is thin. It allows it to slide inward. Thanks to the structural proteins, there's a transmission of force through the entire muscle, resulting in a whole muscle contraction. Now, let's look at the contraction cycle. There's a few things I want you to pay attention to here. If you look at first in the green, right, in the top section here in the green, that's actin. So you're going to see the troponin, the tropomyosin complex. You're going to see calcium. We're going to see the term cross bridge. You're going to see the term power stroke. I also want you to look at the myosin head, and you're going to see ADP and another phosphate hanging out there in the wind. Okay, so let's look at, let's start here on the bottom right. I want you to look at the bottom right. Okay, on the bottom right, we see that there's no attachment between myosin and actin yet. Okay, if you go to the top left, we do see that there's connection. There is a cross bridge between myosin and actin at the binding site. Okay, look at the positioning of myosin. Here's the myosin tail and here's the head. Notice it looks different in this picture compared to this picture where it bent. We see that there's a flick or a power stroke here, or a pivoting that took place. Notice the direction of these arrows here, right? These arrows moved actin in this direction. Well, what happened? Look at ADP and this phosphate. So ADP is adenosine. So let's say here's adenosine, diphosphate, one phosphate, and there's another phosphate. And next to it, there's a third phosphate right here. We're going to put that in this little circle here, connected. So they're kind of next to each other. When it made this contact, it released ADP and this phosphate. Now, usually this phosphate is CP, creatine phosphate, all right? So wherever we see this phosphate by itself, let's refer to it as creatine phosphate, or CP. So it released ADP and creatine phosphate. When you have this adenosine diphosphate with creatine phosphate next to it, this additional phosphate can hook up here to bind and create ATP, adenosine triphosphate. This is monophosphate, this is diphosphate, and here's the third, adenosine triphosphate. That triphosphate is used to detach the myosin head from the actin. Okay, ATP attaches to myosin and myosin head detaches. We know this. This is one of the things that you knew. It's just one of the things you didn't know that you knew, because if you've ever watched any of these like horror movies, or you watch like CSI Miami or CSI New York, any of those where you have the crime scene investigators come to a crime scene and they see a dead body and they see the person in this shortened, contracted state where the elbows are bent, the body's contracted, the fingers are bent, the shoulders are flexed. They're in this shortened, contracted state because remember, when you're dead, you can't produce ATP. and it takes ATP to detach the myosin heads from actin to make the muscle relax. So they're still in this short and contracted state. We just call it rigor mortis. Rigor mortis. Okay? So eventually that ATP, after it removes itself, right? After it's used to remove the myosin head from actin, it goes back to ADP and a phosphate again. Okay, so we're going to take a look at muscle metabolism because that last slide comes into, we're going to connect the dots here because how do muscles derive their ATP? Well, one of it is from CP, creatine phosphate. Another is from glycolysis, anaerobic glycolysis, and then we have cellular respiration. So CP, creatine phosphate, when the body has too much ATP, okay, when the body has too much ATP, it doesn't like it too much. So it's going to store it as ADP and creatine phosphate, okay? Now, if these two connect, if you add one phosphate to ADP, which is two phosphates, you get ATP again. All right, and that's what's happening here. When you have creatine phosphate joining ADP, well, once again, you can get ATP for that. And there's an enzyme that we use called creatine kinase, right? ACE is an enzyme. And that's going to catalyze the transfer of the phosphate group from creatine phosphate to ADP. So here's creatine phosphate. Here's ADP. It's going to require this enzyme CK, creatine kinase, to produce ATP. We need ATP for energy to do any type of work. Okay? The creatine phosphate can provide energy for us, but we use it pretty quick. Like if I'm doing a power lift, if I'm doing a, I don't know, let's say a deadlift, one deadlift, I can use creatine phosphate, right? That's why a lot of athletes, especially power lifters that need short burst of activity, supplement with creatine. The more creatine, the more phosphates can attach. You get like quick, fast burst of energy. So we use this up pretty quick. So then the body can shift gears, okay? The body can shift gears and we can get energy in by a few different places. One of them is cellular respiration. Now in cellular respiration, if we have a cell, we remember we have a mitochondria of the cell, and there's lots of other organelles, but glucose, remember glucose I drew early in a previous video, is a 6-carbon sugar. When we break it down here, we end up with 2 3-carbon sugars. 1, 2, 3, 1, 2, 3. Right? So here, glucose is a 6-carbon sugar. 1, 2, 3, 4, 5, 6. That's glucose. Glycolysis just means splitting the glucose. So you're going to split it into pyruvic acid, which is 2 3-carbon sugars. That's called glycolysis. Now you can make some ATP from that, but not much. And glycolysis takes place in the cytosol of the cell. It does not take place in the mitochondria. What happens in aerobic respiration, if oxygen is available, pyruvic acid can enter the mitochondria, and that's where the Krebs cycle takes place. And the Krebs cycle needs a lot of your B vitamins, B1, B2, B3 for energy. It needs lipoic acid, right? But the Krebs cycle is going to make these coenzymes, and the coenzymes are used by the electron transport chain to produce lots of ATP. So we need oxygen for pyruvic acid to enter the mitochondria for the Krebs cycle, also known as the citric acid cycle. When the Krebs cycle churns, it gives off an exhaust called CO2. And that's what we exhale. So we breathe in oxygen and we give off carbon dioxide, which is given the byproduct from the mitochondria. And the coenzyme H and FADH, these are coenzymes. These FADH and NADH are used by the electron transport chain to produce lots of energy. The electron transport chain right here is dependent on iron. That's why when people are iron deficient or iron deficient anemia, they have no energy, they're fatigued because they can't produce ATP. It also requires CoQ10, coenzyme Q10. The electron transport needs CoQ10 and iron to make energy. It also needs magnesium to produce. Okay, so it's really dependent on lots of nutrition. So this is where and how the body makes ATP. So you need glucose, you need oxygen, you need your B vitamins for the Krebs cycle. It gives off CO2. You need iron, CoQ10, magnesium for the electron transport chain, but the ETC, the electron transport chain, is really making the most ATP, the most energy. But at least you can understand why, if you're iron deficient, why people end up with fatigue. But also CoQ10, this is depleted by people that take statin drugs like simvastatin, Lipitor, Crestor. These cholesterol-lowering medications can deplete coenzyme Q10, and that impairs the mitochondria from producing ATP, and whatever needs ATP is going to cry in pain because they're not getting it, especially muscles. And the term algia, ALGIA, if you attach it to, let's say, myalgia, that means muscle pain. Neuralgia means nerve pain. Have you ever heard of fibromyalgia? Okay, that means muscle pain. One of those triggers could be the statin drugs depleting CoQ10, where there's not enough energy production. Let's take a look, and I will show you how some medications create a drug-induced nutrient depletion that can create a lot of mitochondrial dysfunctions. And I'm going to show you when we come back in another video. To be continued.